All right, so section 2.3 deals with carbon-based molecules that we also call macromolecules. So the reason carbon, or we are referred to as carbon-based life forms, is because carbon is that backbone of the scaffolding of all our macromolecules. So carbohydrates, proteins, lipids, nucleic acids are all built off a skeleton of carbon. That is due to carbon and its very unique covalent bonding patterns. It can bond with up to 4 other atoms because carbon has 4 electrons in its outermost shell, and we've been saying that atoms want to be stable, and to be stable they have to have a full outer shell. So carbon needs 4 more electrons to do that. That means it's not weak enough to give away 4 electrons, but it's also not strong enough to pull 4 electrons from anything else. So it will share. And so, since it does that, carbon can actually form 3 basic types of structures. 3 basic structures: a straight chain, a branched chain of carbon, or a ring structure of carbon. And now all of these structures will have side groups and side chains and other things branching off of them, but the basic formation comes from carbon. So as you look at these structures below, right, it's showing you the straight chain, the branched, and the ring, and you can see that there are other things attached to it, but the backbone is carbon. So these carbon molecules, these carbon macromolecules, are actually made up of lots of smaller subunits bonded together. The process that creates those subunits is what we call a dehydration reaction, because what we do is we take monomers, or what our cells do, our bodies do, is they will take monomers, which are the individual subunits. So think of that as individual Legos, and they will chemically bond them together. And when they do that, they create a polymer. So many monomers linked together gives you a polymer. And remember, poly means many. So we're linking together our monomers, and as we do that, to put them together like that, the dehydration reaction pulls out a water molecule, an OH from one and an H from the other. And when they do that, by removing that, it creates a bond between them. And so by taking out a water molecule between polymer or monomer 1 and monomer 2, you actually create a bond between them, linking them together. So through that process of polymerization, which is a fancy word that means make polymers, through that process we will make our 4 types of macromolecules, or carbon-based molecules. First one are carbohydrates. They are made up of C, H, and O, carbon, hydrogen, and oxygen. It is always in a 1:2:1 ratio for the most part, unless we're talking about polysaccharides. But you can look at this structure and you can see that there are 6 carbons, there are 12 hydrogens, and there are 6 oxygens. A simplified structure would look like the pentagon on the right, or the hexagon, sorry, on the right. The points always reference a carbon. So carbohydrates are sugars and starches. Our simplest sugar are monosaccharides. So mono, again, 1. Saccharide is referring to sugar. So our simplest sugars are monosaccharides. Examples of monosaccharides would be glucose, fructose, which is the sugar found in fruits, galactose. And then when you take those monosaccharides and put them together, you get a polysaccharide. And those would be things like starch, right? Starches that make up pastas and breads, cellulose, which we find in the cell walls of plants, and then glycogen. Glycogen is stored long-term sugar in our bodies, and we store that in our liver. Okay. They can be broken down. One of the main jobs of carbohydrates is for energy. So monosaccharides and disaccharides and polysaccharides can all be used for energy production. Polysaccharides providing the most long-term energy, but they're the longest to break down. They require the most time, whereas monosaccharides are very quick energy, but they don't last long. Then you have carbohydrates that are also helpful in cell structure, like the cellulose in a cell wall, right? So starch is a polymer of glucose, and it's sometimes branched. Cellulose is also a polymer of glucose, but it's a straight chain, not branched. And so here again, structure indicates function, right? The starch is a branched chain with a different kind of bond, and the cellulose polysaccharide is a straight chain with a different kind of bond between them. Second category of macromolecules are lipids. Lipids have a special characteristic. They are non-polar, which means they don't dissolve in water. They are fats, oils, and cholesterols. They contain long hydrocarbon chains. So whenever you have a bunch of carbons attached to hydrogens, we call that a hydrocarbon. In lipids, specifically, we call those hydrocarbon chains fatty acids. So when you look at lipids, there's one very large group of lipids called triglycerides. So fats and oils contain, that category of lipids contain a type of lipid called a triglyceride. Maybe you've heard of that, maybe you've heard it referenced in like blood tests, etc. But triglycerides are basically a glycerol backbone, and glycerol is kind of a sugar, actually. And then it has fatty acid chains attached, like that. So the large orange backbone on the left would be the glycerol, and then those three long tails are the fatty acid tails that are the nonpolar carbon-hydrogen bonded chains that make lipids insoluble in water. They do a lot of different things for us. Lipids, I think you probably know that they are long-term energy storage, so they can be broken down. Fat can be broken down for energy. Also very important in cell membrane function. They make up, we'll be talking about phospholipids and what they do. This is an image of a cell membrane, and all those pink circles with tails there are phospholipids, and those are special kinds of lipids. Those fatty acid tails that you see hanging there are still nonpolar and insoluble in water, but that circular head region there is actually polar. It has a charge and it's attracted to water, which is why when you take these phospholipids and you put them in water, they flip into a bilayer, putting the tails on the inside, getting away from water, and the heads on the outside because they're attracted to water. And so we'll talk about that when we get to our cell membrane and cell transport chapter. Another job of lipids is that they make hormones. So fat is related to hormone production in our body as well. Different types of fatty acids in fats and oils. Those long chains can be saturated or they can be unsaturated. If they are saturated, if you look at this picture, saturated fats have a fatty acid tail where the only kind of bonds you see are single bonds between the CH2s, right? You see all those CH2s, every bond between them are single bonds, but an unsaturated fat has double bonds between those. And so if they even have one double bond, it's considered an unsaturated fat because there's not a hydrogen at every possible, there's not a single bond at every possible location. And so, what's the difference there? Well, unsaturated fats, even though this picture doesn't really show it, they tend to kink, and that's going to cause them to spread out more. And if there's more space between the molecules, they can move more. And so more molecular movement means they're going to be the liquid. Whereas saturated fatty acids are solid at room temperature. Saturated fats come from animals, and unsaturated fats tend to come from plants. Then I already mentioned the phospholipids. Phospholipids are going to make up our cell membranes. They're special because they have two regions, whereas most lipids are just all insoluble in water. Phospholipids have a polar phosphate head, whereas the tails are nonpolar. So one end of it's attracted to water, and the other end is not. And then we get to proteins. Proteins are the polymer, and they're made up of amino acids. There are 20 different amino acids that we use to make the proteins. And we might, estimates vary from 50,000 proteins to more in our body, 50,000 different types of proteins. And all of those are made from the same 20 amino acids, just in different arrangement, different order, different amounts. Kind of like we have 26 letters in our alphabet, and all those letters make up all the words that we have in the English language, kind of that same idea, okay? So those 20 amino acids, and that structure right there is an amino acid. The stuff that is always the same would be that NCC backbone. Whenever you see that structure, the bottom part of it is always going to remain the same. You're going to have an N on the left, a carbon in the middle, and a C on the right, okay? And then that little H atom by itself at the bottom. The stuff in red always changes. There's different types, and that stuff in red is what makes the 20 different amino acids be different, okay? That's what we call R groups, right? So that stuff at the top that was in red on the other slide is now just labeled as an R group. So that varies and changes with amino acid. When we're linking amino acids together, we will do so, excuse me, we will do so by peptide bonds. So amino acids get linked together to form a protein by a certain type of bond called a peptide bond. Okay, so there are 2 peptide bonds linking together 3 amino acids. And again, they vary by the number and order of amino acids. And so those amino acids, when they get linked up together in a chain, are going to interact differently. And their interactions are what create the different shapes that proteins take. And the shape of a protein is super important to what it does. Okay, so all those amino acids that are linked together interact differently to create the shape of the protein. And the shape dictates the function. If you have an amino acid that is placed incorrectly, that's going to change the structure of the protein and the job of the protein. A good example of that is sickle cell anemia. Regular hemoglobin has a certain shape. Hemoglobin carries oxygen on red blood cells. And when it's doing its job, that's fantastic, carries oxygen, everything's great. If you inherit a mutation in the gene for your hemoglobin that causes sickle cell anemia, that mutation is a single amino acid that's wrong. So all those other 300 amino acids are right, but one is incorrect. And that one incorrect amino acid causes hemoglobin to be misshapen, and which then therefore causes the red blood cell to be misshapen and leads to the myriad of symptoms that sickle cell anemia causes for people.